This is our first lecture on cellular respiration and metabolism. And so what is the purpose of cellular respiration? Okay, well, the purpose of cellular respiration is to make ATP, or adenosine triphosphate. And cellular respiration is basically the respiration that goes on inside cells. And it's all about how cells break down molecules, get the energy out of those molecules, and use it to synthesize ATP. So what is ATP? Well, ATP is the universal energy carrier. And in this chemical reaction, we're looking at the hydrolysis of ATP, which releases energy. So let's find the ATP molecule here. So we've got adenosine triphosphate. And so here we are. Let's circle it. And adenosine triphosphate is actually a nucleotide. It is actually the A nucleotide of RNA with a couple of additional phosphates attached to it. So we've got the adenine, shown in green, which is the nitrogenous base. We have the ribose sugar shown in pink. And we have the 3 phosphate groups, which are indicated by the 3 P's that are circled in yellow. And this is our ATP, or energy carrying molecule. And most of the energy is right between the last 2 terminal phosphates. And so in the hydrolysis reaction, remember a hydrolysis reaction, we are adding water, that's the hydro, and using it to break something apart, that's the lysis part of the reaction. So when ATP is hydrolyzed, ADP, adenosine diphosphate, is formed, and this terminal phosphate is removed with the release of high amounts of energy. Okay, so that energy is then used to drive ion pumps, to power muscles, and for many of the cellular reactions that occur in the human body. And so the starting material is the ATP that we need, that we are going to be making in cellular respiration. So this lecture is all about how we make this molecule. So the making of ATP in cellular respiration is part of what's considered to be your metabolism. Okay, in your metabolism, food breakdown is going to lead to the release of energetic electrons. So here's the food that you eat. And obviously you eat it, you digest it, the molecules get absorbed into your bloodstream, and they get taken to all of the cells of the body. And then the food molecules are broken down in the cells, and energetic electrons are released. And when those energetic electrons are released, they are captured by a molecule called NAD, or nicotinamide adenine dinucleotide, but you don't have to remember that. And the transfer of electrons is indicated by the addition of a hydrogen. And so remember that a hydrogen atom is just 1 proton and 1 electron. And so often addition of electrons occurs by the addition of a hydrogen atom. And so the next step here is that the electrons are then used to drive the synthesis of ATP. Okay, so this happens in your metabolism. So what is metabolism? Metabolism is all the reactions in the body that involve energy transformation. Okay, so that is not just food breakdown. Okay, but food breakdown is part of it, because in food breakdown, you are breaking molecules. And when you break molecules, the energy in the chemical bonds is released. Okay, and the breakdown of food is called your catabolism. Your catabolism is your breakdown reactions. But an equal set of metabolic pathways that happens is called your anabolism, because those molecules that are broken down, they also, in many cases, are going to be used to build body parts. And basically, building back up parts of the body is called anabolism. So sometimes you hear that anabolism plus catabolism equals metabolism. So cellular respiration can either be anaerobic, meaning without oxygen, or can be aerobic, meaning with oxygen. In aerobic respiration, we're producing about 20 times more ATP than in anaerobic respiration. And these runners, depending on how far they're running, may be either using aerobic or anaerobic respiration. For example, if they're just doing a short sprint, they're just going to be using anaerobic respiration, which is allowing us to have short bursts of energy. But on the other hand, if they're going to do several laps around this track, they are going to need to use aerobic respiration. Most likely they will be going a lot slower if they're doing several laps around the track. And then you should realize that cellular respiration is not a simple chemical reaction. It's actually a series of metabolic pathways. Okay, a metabolic pathway is a series of enzyme-controlled reactions that leads to a product. In our case, the product is going to be ATP, the ultimate product. But in a metabolic pathway, each new substrate is the product of the previous reaction. So if we look at this picture here, we see substrate 1 is converted to substrate 2. And substrate 2 is actually the product of the first reaction. And this reaction is catalyzed by enzyme A. And so substrate 2, being the product of the first reaction, is also the substrate of the next reaction. And in the presence of enzyme B, substrate 2 is converted to substrate 3, which is also the product of the second reaction. And so when a substrate, or the product of one reaction is the substrate of the next, what happens is you have a biochemical pathway, which is also known as a metabolic pathway if it's involved in metabolism. And some of these reactions may be endergonic, meaning they take in energy, and other reactions are exergonic, meaning they give off energy. Okay, let's look at an overview here of cellular respiration and really pick apart the different aspects of this slide. Okay, so first of all, in this slide, we're looking at glucose here from the digestive tract. And these words here are in the bloodstream or in blood plasma. So the digested molecules have been absorbed into the bloodstream, and also glucose in the bloodstream is also coming from the liver. And this is due to the breakdown of glycogen in the liver. So remember that glycogen is the polysaccharide made of many glucoses linked together. And so then the glucose travels in the blood plasma, and it is taken into cells, and then it is going to be used as a substrate in this anaerobic pathway called glycolysis, which happens in the cytoplasm of the cell, glucose is going to be broken down into 2 molecules of pyruvic acid. And pyruvic acid is a 3-carbon sugar, and glucose is a 6-carbon sugar. Okay, and then if no oxygen is available, the cell will undergo anaerobic metabolism and convert the pyruvic acid to lactic acid. Okay, and this may happen in skeletal muscle if you are really sprinting like those runners you saw in the previous slide. You don't have enough oxygen, and anaerobic metabolism is going to convert pyruvic acid into lactic acid. We'll look at that more specifically as to why lactic acid forms. On the other hand, if oxygen is available, so let's just put plus oxygen gas here, pyruvic acid will travel into the mitochondrion and will break down in a series of reactions called the citric acid cycle or the Krebs cycle. And when the carbon bonds of these carbohydrates are broken apart, electrons are released and they're carried to the electron transport chain, which happens in the inner mitochondrial membrane. And in the presence of oxygen, so through aerobic respiration, ATP will be made with the release of carbon dioxide and water. And so this is where the carbon dioxide comes from that you are going to be breathing out. So let's pick apart the steps of cellular respiration a little bit further and back up and talk in a little bit more detail about glycolysis. Okay, so glycolysis starts with glucose. Okay, and glucose is a simple sugar, and glyc means sweet, and lysis is a loosening or breaking apart. So glycolysis is really about the cleavage of sugar, and as I mentioned, it involves many enzymatically controlled steps. And here you have glucose, which is C6H12O6, a 6-carbon sugar. And these atoms are held together by covalent bonds. And there you have a picture of glucose. And by now you should be able to draw this molecule. You should have a pretty good concept of what glucose looks like. Okay, but in glycolysis, what's going to happen in the lysis part is that one 6-carbon glucose molecule is going to be oxidized to form 2 3-carbon pyruvic acid or pyruvate molecules. And you can use the term pyruvic acid, or you can talk about pyruvate, and pyruvic acid is really the reduced form of pyruvate. So what do we mean by this term oxidized? Okay, so we talked about that in a previous lecture, but let's just quickly review. So we're talking about glucose is broken down, and so we say that it's oxidized. Okay, so we're talking about an energy transfer between molecules, or a transfer of electrons, which are transferring the energy between molecules. And so we're back to this picture that we saw in a previous lecture. Okay, so in this lecture, or in this picture, we're looking at molecule A gives up electrons. Okay, so molecule A has the electrons. When it has the electrons, it's said to be reduced. When it has lost the electrons, it's oxidized. And so that's what's happening to glucose. And in the case of glycolysis, where are the electrons going? Well, we're starting with NAD, as we mentioned, and NAD is picking up the electrons, and now it's going to be NADH. So NADH is basically reduced NAD. NADH has the electrons that glucose used to have. Okay, so molecule B, receiving the electrons, is now reduced. Okay, so cellular respiration is really a series of reduction and oxidation reactions, ultimately breaking down glucose to get the energy out in the form of electrons. And then that energy is eventually going to be transferred to the inner mitochondrial membrane, where we will see ATP synthesis happening. That's in aerobic respiration. Okay, so let's just continue on with glycolysis. And here we have a picture from the book that is talking about the steps of glycolysis. Okay, so our book talks about glycolysis occurring in 2 stages. In one stage, you have an input of ATP energy. Okay, so that's stage 1. We start with glucose, and then we have to actually add a couple of ATPs that you can see are broken down into adenosine diphosphate, or ADP. So we're actually doing an input of energy in step 1. And then in step 2, the energy is released either as ATP or as electrons. So let's look here in stage 2. And so in step 2 of stage 2, we have energy released as electrons, which are going to NAD to make NADH. And in step 3, we have energy going in to make a total of 4 ATP. And ultimately, glucose is broken down into pyruvic acid. On the next couple of slides, we're going to take a look at these 2 steps again, except that we're now going to look at the actual, well, certainly renditions of the actual molecules that occur in glycolysis. Okay, so let's take a look at the energy requiring steps of glycolysis. You recall from the last slide that we had an input of 2 ATP in the energy requiring steps. Okay, so glucose is broken down into glucose 6-phosphate. So you can see that a phosphate from ATP is actually added on to the end of glucose to form glucose 6-phosphate. This molecule then rearranges to form fructose 6-phosphate. But you know that we need another ATP input at this stage. And here it goes. ATP is going in, and another phosphate is being added to the sugar, which was now fructose 6-phosphate, to become fructose 1,6-bisphosphate, or sometimes you hear it called diphosphate. In other words, there are 2 phosphates now onto this sugar molecule. And then fructose bisphosphate is then energized to break apart and form 2 molecules of pGal, or phosphoglyceraldehyde. And then one of the things you should realize about the phosphorylation of glucose is glucose 6-phosphate cannot cross the cell membrane. So this reaction sort of traps glucose inside the cell. Okay, so only if a cell has an enzyme that converts glucose 6-phosphate into glucose, can glucose then get back out of the cell again. And that's going to become important when we compare skeletal muscle versus liver. Let's go on to the second set of reactions, which are the energy releasing reactions of glycolysis. And so we're going to start off with these two molecules of pGal. Okay, so now each of these molecules of pGal is going to rearrange themselves to eventually form two molecules of pyruvate. And so let's just look at these various steps. First of all, pGal phosphoglyceraldehyde is converted to 1,3-bisphosphoglycerate. A phosphate is added on to the opposite end of the pGal molecule, and electrons are released and added to NAD to form two molecules of NADH. But then one of those phosphates comes off and 3-phosphoglycerate is produced with the release of 2 ATP. 3-phosphoglycerate is then converted to 2-phosphoglycerate, and with a release of water is converted to PEP, or phosphoenol pyruvate. And phosphoenol pyruvate, which is also phosphorylated, then releases a phosphate to form ATP. And of course there are two molecules, so 2 ATP are formed. So in these reactions, 4 ATP and 2 NADH are produced. Okay, so if we look at glycolysis on the whole, we see that 4 ATP are produced, but there was an input of 2 ATP. So we say we have a net gain of 2 ATP molecules in glycolysis. And we also have a net gain of 2 NADH. Next slide, we have just a little review for you. So what is the purpose of NADH here? Okay, so let's just take a look here. We've got the exergonic reactions of glycolysis releasing electrons, and where do they go? Okay, so they go to NAD to make NADH. Okay, and then NADH is eventually going to release those electrons to drive an endergonic reaction, which will be about the synthesis of ATP. So NADH receives electrons in a redox reaction. And FADH is also another electron carrier. It works just like NADH, and we're going to hear about that when we talk about aerobic cellular respiration in the next lecture. But in glycolysis, we've made two molecules of pyruvic acid. And what happens in anaerobic respiration? Well, in anaerobic respiration, those molecules of pyruvic acid are going to be used to form lactic acid. And that's what's shown here. Pyruvate is converted to lactic acid. Okay, so let's just take a look here. We've got glucose, so now we're in glycolysis, and we get a net gain of 2 ATP molecules. We're just showing this simplified glycolysis reaction. We know from our previous slides that it's actually many more enzymatic reactions. So glucose is going to become 2 pyruvate molecules. 2 ATP are formed, and 2 NADH are formed. Okay, and in lactic acid fermentation, so when we're exercising and we don't have enough oxygen to make a lot of ATP, rather than going into the mitochondria and driving the Krebs cycle, pyruvate is going to be converted to lactate or lactic acid. And since we have 2 of these, they will be converted to 2 lactates. And we have the 2 NADHs from glycolysis, and those are going to be used to reduce pyruvate to make lactate. And that means the NADH is then oxidized and NAD plus is then resynthesized. Okay, so what is going on here? Well, first of all, what's going on here is that your body doesn't have enough oxygen to make a lot of ATP, so it's going to try to make ATP this way. But if you look at the chemical reaction in which pyruvate is converted to lactate, actually there's no ATP formed. What's happening is you are regenerating the NAD, which can be used to capture the electrons during glycolysis, so that you can get 2 more ATP released in glycolysis. Okay, so the benefit of lactic acid fermentation is that NAD plus is produced, which can be used for glycolysis to help make 2 ATP, which is just a little ATP. Okay, now I have on this slide that lactic acid causes soreness in skeletal muscle, but you should realize that there is still quite a bit of debate around this. And, there's some suggestion that lactic acid is able to cross into the mitochondria in the presence of oxygen. And there's a lot of information showing that lactic acid is the one of the main sugars that is required for brain activity, in specifically memory. And so when we talk about muscle soreness or muscle fatigue, realize that the soreness may be due to a variety of factors. One which may simply be the release of hydrogen ions from a source other than lactic acid. But also realize that your skeletal muscle is going to be able to use lactic acid fermentation to respire anaerobically, but the heart only respires aerobically. Okay, so you have to have oxygen for heart muscle. And red blood cells, on the other hand, only use lactic acid fermentation because they lack mitochondria. Remember that one of the steps on the developmental pathway to make a mature erythrocyte is the ejection of nuclei and the other organelles. So red blood cells don't even have mitochondria. So what happens in the body after you stop exercising to the lactic acid? And so after you stop exercising, you start to have an intake of more oxygen. And so there's something called oxygen debt, which is the amount of oxygen you need to convert the lactic acid back into pyruvate. And indeed, that is what happens. And so, but of course, this requires an input of oxygen. And so this happens in the Cori cycle. Okay, and lactic acid is going to be in the bloodstream. It is released from skeletal muscle and is converted back into pyruvate in the liver. And so let's just look at these reactions in skeletal muscle during exercise. You have the breakdown of glycogen, which can be stored in skeletal muscle to form glucose 6-phosphate. Okay, and that was the first step in glycolysis. And then, of course, glucose 6-phosphate is eventually broken down into pyruvate, which is used to make lactic acid. Okay, and that's an anaerobically respiring skeletal muscle. And lactic acid, we're just following the numbers here, is released from skeletal muscle out into the blood and goes to the liver. And in the liver, there is an enzyme called lactic acid dehydrogenase, which converts lactic acid to pyruvic acid and then back into glucose 6-phosphate. Okay, what's the benefit of this? Well, first of all, glucose 6-phosphate in the liver, if glucose is not needed in the bloodstream at that time, glucose 6-phosphate can be converted into glycogen for storage in the liver, and then glycogen can break down into glucose 6-phosphate again in the liver. And that's good because liver has the enzyme glucose 6-phosphatase, which breaks glucose 6-phosphate down into glucose. And glucose can then be released back into the bloodstream, and it can go to all the cells of the body, specifically skeletal muscle can be taken into skeletal muscle and then used to make glucose 6-phosphate again. And when you are at rest then, and you are in an anabolic state, glucose 6-phosphate will be used as a precursor to make glycogen for storage again in skeletal muscle. But the important thing here, and this is known as the Cori cycle, one of the important things here is that the liver can convert pyruvic acid to glucose 6-phosphate and then into glucose. Okay, and glucose can be released into the bloodstream. But if you recall my saying that glucose 6-phosphate itself cannot be released out of cells, okay, and so it's trapped inside the cell. So the liver is the organ that has the enzyme that can convert glucose 6-phosphate to glucose, but skeletal muscle does not have that enzyme. And so glucose, it's relying on blood glucose or the breakdown of glycogen stores for a food source. And then finally, on the last slide, I'd like to go back to talking about the heart, because we said that the heart cannot do anaerobic respiration. Okay, so what happens if there is not enough oxygen going to the heart? Okay, and students should be aware of the clinical application, which is certainly in my edition of Stewart Fox, edition 14. And so if you don't have that edition, you might want to get a hold of that from a friend or ask me during class if you can read. It's just a paragraph to read about. And also this slide will kind of summarize what's in that clinical application, which is about myocardial ischemia. And this refers to inadequate blood flow to heart muscle. Okay, and ischemia is really about a lack of oxygen. And so when we're talking about ischemia oops, I just realized this is spelled wrong. This should be myocardial, C-A-R-D-I-A-L, not myocardial. Okay, so in myocardial ischemia, this is sometimes called coronary heart disease because of an inadequate blood flow in the coronary arteries. Remember, those are the arteries that are feeding the heart directly. And is also called atherosclerotic heart disease because the coronary arteries in atherosclerosis are filled with plaques that are made up of cholesterol and fats that are basically preventing blood flow through these vessels. And so oxygen can't get to the heart. And prolonged ischemia can cause the death of heart cells, which ultimately will lead to myocardial infarction, which is kind of a general term for heart attack. And then, but realize that if lactic acid builds up in heart muscle, patients will get chest pain, and this is referred to as angina pectoris. Okay, and this is what they use nitroglycerin to treat, okay, because the function of nitroglycerin is to improve blood flow to the heart, which will then reduce the work that the heart is doing by dilating the peripheral blood vessels as well. Okay, so that ends our first lecture on cellular respiration. And in the next cellular respiration, we'll get much more into aerobic cellular respiration.